Thank you so much, Dr. Murthy. All right. What time is it now? It's good afternoon. Thank you so much for coming to this wonderful conference, and I'm delighted to be part of this. And I'm looking forward to having a wonderful session with you in the next few minutes. So Dr. Mishra talked about translational research, and he was heavy on translational medicine, and you learned a lot about what is going on the translational side. I will try to focus on the molecular medicine side for my portion of the talk, okay? And today, whatever I'm going to talk today is unpublished data. So the idea is that we will learn something new, in this conference. Let's hope it works. All right, as I was saying that we will talk about something new, and I hope that we will learn something new in this talk. And this is more directed towards students, so I'll be very lay in my talk. Although you will see some data, we may not go too much into data. The idea is that we will try to learn the bigger picture, okay? All right, so we will talk about drug resistance in cancer. We will talk about some newer therapies and newer technologies that we are working to develop, and a new cell death mechanism that we think we have identified. So we will be learning about a few different aspects in pharmacology and especially molecular medicine, and you will learn how we are approaching towards developing a new class of drug now. Okay, so I'm from University of Toledo, which is on the border of Michigan and Ohio, and you will see a few other professors from University of Toledo today. It is a large program with 450 different programs, and spread into a few different campuses. I'm on this right-hand side. Okay, this is not working here. Okay, we'll just continue with the slide then. Okay, so I'm in this building where my research lab is located. Okay, so I run this program, Cancer and Systems Therapeutics, and I wear multiple different hats. My research work is in the area, as I said, on drug discovery, drug design and development, more focused toward multi-drug resistance, right? But we do heavily precision medicine. We design and develop drugs for a number of targets, both from synthetic sources and natural products. Not going to go too much in detail on that. Other area of interest is molecular biology. So I'm very interested in transporter biology, for example. A portion of my lab is now also working on anti-aging biology. And then we are very interested also in developing new devices. So you will see some posters outside which is printed, and we have come up with a few devices that we are working on. And I'm very eager that if you have any questions, we'll be happy to talk about those. What I really want to talk today, as I said, is something very new in my lab. And again, it's not published, so your feedback would be very useful to move this area forward. So we will learn about something that I think is very interesting. But before we do that, let's get back to basics so that we are on the same page. All right. We'll start all the way from cancer, and everybody knows cancer is the second leading cause of death. There are a number of treatment modalities, from surgery, radiation, as you learned from Dr. Mishra, then chemotherapy, and now immunotherapies. So chemotherapy and immunotherapies are still considered as a major modality. However, what happens in patients is that over a period of time, they stop responding to these drugs. Not only the drug that they are being treated with, but other class of drug which are structurally and mechanistically unrelated. We call it multi-drug resistance. समझ में आ रहा है कि हिंदी में बात करें? क्योंकि अगर आप नहीं समझ में आ रहा है तो I will be very happy to talk in Hindi also. But what I will do is कि जहाँ पे मुझे लगेगा कि difficult concept है, I will switch to Hindi for you, okay? So this is a very important area. Multi-drug resistance is very, very complex area to work, because when the drugs don't fail to work, we have no treatment option. Okay, you have to just give more drug to and hope that will work, and that is not always a good strategy, because that also leads to toxic effects, right? So you switch to other drugs and nothing works. So multi-drug resistance poses a lot more problem. So if you talk to oncologists, they will say that almost very few patients, if they do, they die from a localized tumor. Tumor, when it is localized, it's very easy to treat them. It's when the tumor actually spreads from one part of the body to the other parts of the body, we call it metastasis, that's when the problem occurs. And we now have come to believe that most of these tumor metastases occur when the tumor stops responding to these chemo or immunotherapies or some other radiotherapies, right? We call it drug resistance. So drug resistance in part leads to metastasis, can lead to poor prognosis and survival in patients. Okay, so that's something, that's why it's very, very important to target multi-drug resistance. So before we do more, we should learn about what multi-drug resistance is, right? So as you know, drug needs to be absorbed and it needs to be distributed. It needs to target site, which is cancer cells. And in cancer cells, we are trying to kill them, right? So these drugs enter the cancer cells, they go and bind to its target, they change whatever conformation changes it has to do, whatever signaling pathway it has to conduct. And in most cases, they kill the cancer cell through a cell death mechanism called apoptosis. Apoptosis, as you already may know, it's a programmed cell death, right? So once it has done its job, it has to be excreted or it's metabolized, and then it is removed from the system. But oftentimes, if the drug is not properly absorbed or not properly distributed, or even when it reaches the target site and there is mutations in the target site or there is conformational changes in the target site, it would not be able to do its job. Or if it gets metabolized inside the cell, you have to give more drug to get the same effect. And there is other thing that is also present in the cancer cells. There are some transporters which are present on outside of the periphery of the cells, on the cell membranes. We call them efflux transporters or ABC transporter. They can pump the drug outside the cell. Or the drug by itself can be metabolized very quickly. So all these factors can lead to drug resistance. Okay. So I was very excited, when I started my journey as a pharmacist. So I did my pharmacy, in India, and I was very excited to be a pharmacist because I thought that drugs are like magic. आपने किसी को एक गोली दी, fever या कोई भी बीमारी है, and they are like treated. इससे ज़्यादा मतलब what more magical there could be, but this magic often fails or fades over a period of time. बहुत सारे patients lucky नहीं होते हैं कि वो terminal condition पे survive कर पाएं, right? It's because of the development of drug resistance, as I said, but also there could be other reasons for chemotherapy failure, such as variable bioavailability, toxicity, side effects, or unpredictable drug-drug interactions. So one of the common factors that we found was ABC transporters, as I was saying it's expressed on the plasma membrane. There are 49 of those. 9 are very important for drug resistance, and we studied them heavily. So you do understand their clinical importance. You will appreciate that almost 50% of all the clinical drugs that are out in the market are substrate of these ABC transporters, which means that ABC transporter can cause drug resistance or pharmacokinetic alterations to almost 50% of the clinically approved drug. These ABC transporters are also present in more than 70% of the cancer patient, which can lead to again, drug resistance in these patients. So they are very, very important thing to target. But I just hope that you understand what they really do in the body. So if you can look at this cartoon diagram, you will understand that they are actually present in the body to protect our body against xenobiotics. So if you know that the drug enters the cell through either diffusion or carrier-mediated transport, but these transporters actually pump the drug outside the cell, and because they think these cancer drugs are xenobiotics, they will pump the drug outside the cell. And so the intracellular accumulation of drug is not enough to kill the cancer cell. So we came up with a very simple hypothesis. Okay, why don't we just simply block these ABC transporter at the ADP binding site? बहुत simple strategy है कि आपने इसको pump को ही बंद कर दो, तो drug will be enough concentrated inside the cell, should be able to kill the cancer cell, right? So we did that, and we spent hundreds of millions of dollars for this strategy by number of different mechanisms to target ABC transporters. We published several papers in top-notch journals, to support our hypothesis that yes, this would be a good strategy. But long story short, there is not a single ABC transporter inhibitor in the market. Okay, there is no FDA-approved drug in the clinic, and I can go on and on and on about why these, transporter inhibition failed, and there are a number of people who have come up with different hypotheses, and of course we have seen that these inhibitors by themselves were toxic. They were changing the pharmacokinetics of parent drug. But one thing that we also realized is that they were at a time targeting only one ABC transporter, which is not a good strategy, because now we know that these ABC transporters are, often co-expressed. If you look at the key organs such as brain, blood-brain barrier, or kidney and liver, they have multiple different transporters that are co-expressed. They also have a similar function. They have functional overlap. And if you look at number of different anti-cancer compounds, more than one transporter can pump the same drug. So if you're only inhibiting one ABC transporter, that may not be a very good strategy. Even making the matter more complex, if you look at the organ level, if at the cell level, if you look and zoom in, at the hepatocyte level or renal level, you can see that these transporters have different orientation. Some pump the drug outside the system, some pump the drug back into the blood circulation. So there is very nice homeostasis. So if you inhibit one transporter, how can you not assume that other transporter will take its role? We call it redundancy of transporter. So we did a lot to understand that, and we are still working to understand how to target this overlapping function of transporter. Okay, and so we used, now we have developed some new agents that can target different transporter at different time. So we can target ABCB1 and BCRP and BCRP and MRP2. So we have number of these transport inhibitors. But again, this is a very, very basic sort of work that we are still working on. But then we realized that ABC transporter is only one small drug resistance factor. Okay, तो कहानी अब यहाँ से शुरू होगी. अभी तक जितनी मैंने भूमिका बनाई, वो सिर्फ एक भूमिका थी दिखाने के लिए कि काफ़ी काम गया इसमें, but I have reached nowhere because nothing actually came to the clinic, which is very painful. अगर आप कुछ काम कर रहे हो और कुछ का result निकल के नहीं आ रहा है, it's good to provide knowledge, and we learned a lot by failing. So that's part of science. But then other thing is you have to zoom out and see कि अब target क्या कर रहे हो. We realize that there is a much bigger drug resistance factor that we are just not targeting, which is apoptosis resistance. 95 to 98% of the clinical drug that are anti-cancer drugs kill the cancer cells through apoptosis. And oftentimes mutation in that apoptosis leads to drug resistance. So why have we not targeting that? And at that point, my lab focus was much towards in that area. So now, I will talk about what new developments we have made in this area, and perhaps I hope that by the end of the talk, you will learn about some new cell death mechanisms and some new strategies to target apoptotic resistant cancers. So again, I'll just bring back to basics. So you already know what is apoptosis, right? In the cancer cells, you give the anti-cancer drug to patients. What happens? The cancer cells, they shrink. Okay, there is membrane clubbing. They then condense. There is a chromatin condensation, there's nuclear condensation. They form these apoptotic bodies, which is then phagocytosed. We call them cell death, suicide, or the programmed cell death. There's a lot of markers that are involved. It's a very regulated process. Right? There could be intrinsic apoptosis or extrinsic apoptosis. Caspases are involved in doing that. Okay. Or they can die through necrosis. If there is infection or there's injury, they can swell and then they can burst and it could lead to inflammation and they can die. So these are the two cell death mechanism that is very well understood. Okay. Now let me talk about the discovery that we made. And it was an accidental discovery because we were not looking at this particular type of cell death. We were actually trying to develop a dual tyrosine kinase inhibitors, and I have some chemists here, Dr. Trivedi's group and Karthikeyan, who was working on developing these dual tyrosine kinase inhibitors, which we thought are fantastic, and there were like 42 such compounds that were synthesized, which were doing what we wanted them to do, and they were binding to the target. But one day, my student came running, and she was screening those compounds, and she says, "Dr. Tiwari, I'm seeing something very strange. Can you come and look at that?" And I went to the lab to see what she's talking about, and I wait for such kind of moment to see something unusual. That's where discoveries are. So what she was seeing was very phenomenal. She was seeing that these cancer cells were filled with vacuoles after she treated these compounds with that. these cells, for some reason, looks like they were drinking themselves. There was too much vacuoles. There were vacuoles forming, they were fusing together, they were becoming bigger and bigger, and then they were bursting. So you can see that how there are hundreds of gunshots in the cancer cells. These are nothing but vacuoles. Okay, very unusual phenomenon. And so when we see something like this as a scientist, we don't right away believe that. So, and I did not, and I asked her to repeat this with multiple different controls, and every time she got the same data, which was phenomenal, and that was, I've never seen something that before. So what we do, we go back to the literature, and that's what we started looking into it. And turns out that just few years ago, Bill Maltese, who's was past chair of cancer biology at University of Toledo, described a new cell death mechanism. He coined that new cell death mechanism as methuosis. What does methuosis mean? Methuo in Greek means that you drink to intoxicate. आप इतना पानी पियो, पीते जाओ, और कोई आपका urinary tract बंद कर दे, क्या होगा? फट जाओगे. Right? That's what we were seeing with these cancer cells. So he developed, in collaboration with my collaborator, a class of compound called MOMIP. Right? So as soon as you give this MOMIP, they will start forming these ruffles. They will take the extracellular matrix. Vacuoles will be formed. They will fuse together. They will form larger vacuoles. They should then go and bind to the lysosomes. Lysosomes are digestive organ in our cells, but instead they were not binding to the lysosome, and they were becoming bigger and bigger. And they were not also forming any autophagy. They were just drinking themselves to death, but they were not eating themselves. Anyways, we thought that, cool, we have found a new class of compounds which has structural, it's very different, and let's focus on developing this new class of compound which may be doing methuosis-like phenomenon, right? So assuming that we worked with my team, chemistry team, in India and in the US to come up with new class of compounds. And the goal was, okay, we are seeing this high IC50 values, can we optimize them to bring it to nanomolar potency? And we came up with that, and we came from BAP13 to BAP27 at that time. Now we have much more potent compounds in the library. We could see that they can form these vacuoles and kill the cancer cells at a very small concentrations, and you can do a bunch of cytotoxicity as assay. And I'm not going to go in any detail on any assay, but just to show you that these are very effective in cleaning and clearing out cancer cells and inhibiting their proliferation. So just so that you understand what this process is, this dysregulated macropinocytosis, what we call methuosis. So as soon as you give the cancer and BAP compound to the cancer cells, this is colon cancer, they will start forming vacuoles within an hour. They will keep forming vacuoles, they will fuse together, then they will become one big large vacuole. It will detach from the substrate, and then they will pop, right? So, I did not put the video because somehow my videos don't, support it in many places, but you can appreciate in the bottom that how these, cells were bursting and the liquid was coming out. And you can do transmission electron microscope to see how these vacuoles were actually fusing and becoming a large vacuole. So we know that there was macropinocytosis going on that was leading to cell death. But that is not enough to convince my scientific people here. They say, okay, this is good, but how do we really believe that? Show me with more supporting evidence. So you have to do more assays, like molecular assays, right? So we did, of course, a lot of electron microscopy work. Then we looked at the tracers where their vacuoles were actually taking in extracellular dyes. Then we looked at Western analysis to look at these biomarkers that are involved in macropinocytosis, where there is endocytosis, is late endosome formation. So RAP7 and RAP5 is upregulated with increasing concentration, as you can see there. In addition, you will do more microscopy work. And then what we saw was very interesting. While we were looking at this macropinocytosis, which we now believe that there was a lot of methuosis going on, these lysosomes were turning very acidic, very red. Then we started looking at our electron microscopy data to understand what is going at the lysosome. So if you zoom in into a cell, you are looking into a very zoomed image of a cell, and you're looking at a lysosome which is 15-fold to 20-fold more swollen compared to regular lysosomes in a cancer cells. There's too much activity going on in these lysosomes. You can see these double membranous structures, as if they are eating a lot of things. I said, wait a minute. they were drinking themselves, methoosis. What is this? I mean, we were not expecting them to also eat themselves. So this was our first observation when we saw that lysosome is very acidic, that they are eating themselves. I said, that is very interesting. So maybe they are also doing autophagy. So as you know, what is autophagy, right? It's a self-eating process where when the cells are very stressed, or there is some changes in their organelles, or the organelles are not functioning, they form these double membranous structures called autophagosomes, which then go and bind to the lysosome. Lysosome is a digestive body. It forms autophagolysosomes, and these, they digest them and produce energy, like different types of amino acids, nucleosides, sugars, and everything. And this biomass is used for cell survival process or proliferation to the new cells. I said, wait a minute, cells, autophagy is leading to cell survival? That is true. If there is normal autophagy in cancer cells, there is a lot of autophagy that goes on, which allows them to produce more of this biomass so that they can survive. But in our case, cells are actually dying. So are they dying only because they are drinking themselves to death, and maybe autophagy is not important for them to cell death or not? So we had to really figure that out. And so we started looking into autophagy. First thing is whether autophagy is happening or not. So we did a bunch of molecular assays, and we were convinced, yes, LZ3B2 is going up, significantly up. That means too much of autophagy was going on. Then we started looking into more molecular mechanism and signaling pathway, which suggested, yes, autophagy was going on, mTOR was inhibited, which is a negative feedback loop of autophagy. But then whether autophagy was leading to cell death or not, you can use molecular probe. So if you give autophagy inducers such as rapamycin, it will lead to more cell death in our hypothesis, and that was the case. Or if you inhibit autophagy by giving inhibitors such as 3-methyladenine, it will lead to cell survival. That's what we were seeing. So that means that autophagy was actually leading to cell death, right? And then you can do more and more and more assays to really prove your point. You have to support your, data with more evidences. You can use 3-chloroquine or hydroxychloroquine to support the same data and the cell survival. It all suggested that autophagy was leading to cell death. So long story short, मैंने आपको अभी तक ये बताया कि we discovered accidentally a class of compounds which was leading to cells drinking themselves, a process called methoosis. काफ़ी सारे bubbles बन रहे थे, vacuoles बन रहे थे, fuse नहीं हो रहे थे, and that was leading to cell death. At the same time, they were making the cells eat themselves, autophagy. मेरे लैब में लोगों ने इन दोनों की शादी करा दी. So methoosis or autophagy की शादी हो गई, and then a new word was born, sort of methooophagy, and that was a common term that we use for this, describing this unique sort of cell death mechanism. So you will hear this term much more now, methooophagy. So once you discover any new phenomenon, we generally don't stop at that. We need to understand what is going on with that. So first thing is whether it is apoptosis or necrosis, right? Apoptosis is where the cells shrink, and 95 to 98% of the drug do apoptosis. So we really have to figure it out. So we did that, and methooophagy is a non-apoptotic mechanism. Why? Because the cells, instead of shrinking, they are swelling. Nucleus is happy and healthy. There's no changes. There is no nuclear condensation. And if you look at the molecular pathways, there is no change in caspase's cleavage. Backspac, which is a apoptosis marker, did not change. If you use pan-caspase inhibitor ZVAD, it did not rescue the cell death. इसका मतलब है कि methooophagy was a non-apoptotic cell death, right? And we did a bunch of other assays to really prove that this is non-apoptotic cell death. Then we looked at other cell death mechanism that has been described in literature. One is called necroptosis. It's a programmed necrosis. So when you give necrostatin along with BAPT compound, it should have rescued the cell death if it were a necroptosis process. But here it did not, which means that methooophagy is a non-necroptosis cell death mechanism. We looked at a bunch of other cell death mechanisms, such as ER stress or mitochondria swelling. We looked at reactive oxygen species. There was no reactive oxygen species generation. We looked at beta-tubulin because there are a lot of anti-cancer agents that kill the cancer cell through beta-tubulin. We did not see any changes in beta-tubulin inhibition. That means there was no beta-tubulin involved. We also looked at RAS and P53 status. Why? Because in colon cancer, more than 50% of the colon cancer has this mutation that leads to colon carcinogenesis, right? And we use a bunch of different models to prove our point. that some are KRAS or P53 mutated, and methylophagy was happening in all of them. That means that P53 or KRAS status does not matter as long as methylophagy is concerned. Okay, so why I was showing you all that negative data? कि यार कुछ भी तो नहीं काम कर रहा है जो कि already proven है, because it is something different, which is it's a non-apoptotic cell death mechanism. तो बहुत सारे apoptosis related जितने भी cell death हैं, it is not behaving like that. So while we were trying to understand cell death, part of my group said, okay, we are here to solve the problem of apoptosis resistance. Is it going to overcome apoptosis resistance? Can we really find out? So as you know, in colon cancer, most of the drugs that are used in colon cancer kill the cancer cells through apoptosis. So you can create apoptosis models. You can take colon cancer and you can knock out different models, and you can make different models. For example, back are pro-apoptosis market. So we did single knockouts and double knockouts, and you can see that when you give 5-fluorouracil, which is used for colon cancer, the curve shifts towards left. You have to give more drug to kill the same cell line, which is double knockout. That means apoptosis resistance. They're resistant to apoptosis. But when you give back compound, that was not the case, which supports our data that it is a non-apoptotic cell death. It doesn't matter what kind of mutation is there in apoptosis. It will still kill those cancer cells. And it was very cool. So we started combining them with other anti-cancer agents. And when we combined them with other anti-cancer agent in colon cancer, we saw synergistic to very strong synergistic data. And oftentimes this is all good. You find a drug which is very, very cool, killing the cancer cells through a non-apoptotic mechanism by making them drink themselves, eat themselves. But is it safe? Can you actually move it forward or not? And we have to study that much earlier, because if you spend so much of energy and effort to understand the whole mechanism and you don't know whether this class of compound is safe or not, it will pose a different problem later on. And what we found in number of different cell models, in mouse models, that's pretty safe. And I worked very closely with Dr. Williams, who's going to talk about zebrafish research, and we looked at, it was pretty safe in zebrafish up to even 100 micromolar concentration. It suggests that they were very safe. We did organ toxicity, we did blood chemistry toxicity, we do a lot of toxicity. I'm one of the board certified toxicologists, American Board of Toxicology. I'm very interested in toxicology early on so that we can save the pain later, right? So we are now working on human, PDX models for colon cancer to understand its efficacy in those models. So you may be thinking that I switched अच्छा खासा mythology के बारे में बात कर रहा था, then I switched to its efficacy and apoptosis resistance, but we never failed to really understand कि हो कैसे रहा है. एक phenomenon दिख तो रहा है कैसा हो रहा है, but हो कैसे रहा है? What is the methylophagy target? And that is often $1 million question. Just to answer that question, it will require us millions of dollars, right? But we started from somewhere, and we have to start somewhere. So we did heavy genomic analysis, and I'm not a very big fan of this analysis early on because it often gives you more answers and more distractions than actual targets. And so we have been talking with Dr. Mishra, and I hope that we can get some radionucleotide to target, and do some binding affinity assays later. But we started with this, and we got some hits, and some metabolic pathways were disturbed. It gives us some direction. Then we did more kinome analysis and looked at some targets. Some targets which we were assuming would be the target for necrosis and autophagy was very, very prominent. PIK5 is one of them, and then there are JNK signaling pathway. So we have an idea what is the cell death pathway. But real thing came in when we went back and looked at our own data. So when we looked at our lysosomes, if you look at lysosomes here, you will see that a lot of autophagosomes, these yellow pockets are nothing but autophagosomes, and these are lysosomal enzymes. What you're seeing here is when the cell is eating itself, a lot of autophagosome is supposed to go to the lysosomes, and lysosome is swelling and becoming bigger and growing more in number. What they should be doing, they should be digesting them and releasing those materials for cell survival. Instead, somehow that lysosomal clearance was blocked. So that is very interesting because there is no such drug that we know can block lysosomal clearance in this way. It was very, very unique sort of understanding at that point. So we started looking at the signaling pathways or homeostasis that controls that lysosome to the nucleus homeostasis, right? That is mTOR TFEB signaling pathway, which did make sense because mTOR was significantly downregulated. That's why autophagy was happening. But TFEB was also significantly downregulated in the cytosolic portion, while it was upregulated in the nuclear portion, which suggests that mTOR TFEB signaling pathway was super dysregulated signaling pathway, but we do not really know the targets yet. We have some idea, and our hunch is that it is a multi-targeted compound, but we're still trying to figure that out. Okay, but so far what I taught you, or at least showed you, or demonstrated is that autophagy is used for cell survival, but when you give BAPT compound, it leads to methoosis. At the same time, autophagy is dysregulated, and both are blocked. This lysosomal clearance is affected because of mTOR TFEB signaling pathway, that both leads to cell death. Okay, that is what methophagy is. So we did not stop here. We continued to optimize our compounds to make it more effective and more potent, and we have come up with a large library of compounds to do structure activity relationship. Now we have a class of compounds that only does methoosis, or can only do autophagy, or can do both methophagy. So we have a very good understanding what kind of chemical alteration needs to be done to do those things, and we are still trying to understand this lysosomal to nucleus trafficking. So this is the entire story of methophagy, which is killing the cancer cells. But while we were on this project, there's another project popped up, on the side. Some of these compounds, when we were synthesizing, were not actually killing the cancer cells. They were actually forming a lot of vacuoles. So that is very cool. So my team came up with very, good hypothesis that antibodies, and we talk a lot about antibodies, right? The biggest problem in antibodies is that they're large, they cannot be delivered inside the cell. Otherwise, we can target a whole, targets inside the cell. Most of them target only outside, on the outside, and that's something we know. And again, this is a very recent sort of a data. So, as you know, large molecule is a huge market, up to almost $720 billion dollar market is going to be very soon. In that, just if you look at US antibody drug conjugate market, it's growing rapidly, right? So very, very cool area. But the biggest problem is small molecule can easily pass, but large molecule have problem delivering, right? Antibody drug conjugate, antibodies are attached to these conjugates. They are taken in, they are endocytosed, or they go through clathrin mediated transport. Once inside the cell, they will release, and these antibodies are digested, and they're not useful anymore. So we started looking into this macropinocytosis pathway, and it is really, was very fancy data that we got, that when we use very large molecule up to 150 kilodalton, they can easily go through along with this macropinocytosis inducers. Okay, how are we doing with time, Dr. Murthy? I can go on and on. And these were actually proving to be very synergistic when you combine with large molecule with these macropinocytosis inducers. So we have a very good idea how to develop this project, but it's in a very infancy. We have some issues with this project. Some of these antibodies, again, even they are taken in, they are not being released effectively. There is some issues with the endocytic clearance, which we are trying to understand. So this is something that is going on. Do we have time for one small project? I will just run very quickly. So this was methophagy, drug delivery sort of thing. I mean, I worked a lot with professors here. Dr. Ramapuram and Dr. Bodhu is here on formulation side of it. You will hear them talk. But one of my area of research is on triple negative breast cancer. I'm one of the Susan G. Komen breast cancer fellow, very, very interested in effectively coming up with treatments for breast cancer. So I work on triple negative breast cancer, where there is 3 receptors absent: progesterone, estrogen, and HER2. That's why they call triple negative. 15% of the population have triple negative breast cancer. They're very aggressive. There's very limited treatment option for those, and we are trying to find out new drugs in that class for those compounds. Again, I didn't put Dr. Karthikeyan picture here, but he's also very involved in that project, and that is also patent protected along with other agents that we just showed. So we came up with a new class of compounds called TPH104. It came through phenotypic screening. They were actually making the cells swell, and they were killing the triple-negative cancer cells. I'm not going to go in details. Here you are seeing that they are very selective. They kill the cancer cells in nanomolar potency, and they're very selective to triple-negative breast cancer. So you do a lot of selectivity studies with normal cancer cells, and you do a bunch of cytotoxicity assay to prove your point. Then you do other molecular mechanisms to understand its molecular, what kind of cell cycle they're attacking, how they're affecting the proliferation. But our goal is to also understand how they're killing the cancer cells, apoptosis or non-apoptosis. And it actually came from the screen of non-apoptosis signaling pathway. So we were assuming that these are killing through non-apoptosis. So if you can see this figure, then you will understand that as you see the cancer cells being treated, with these TPH compounds, and this is triple-negative breast cancer, eventually they will start swelling, and as they swell, they will release this dye, and propadium iodide that is inside will come in, right? So I cannot forward it here, otherwise I would have done it. You have to bear with this video. So as you see, the cells are growing, and as they grow, they pop, and the red dyes will start popping, seeping in, which is propadium iodide. Right now, the cells are dead, and this is a very unique sort of mechanism. It is very necroptosis-like cell death mechanism. That we found through this screen. So we do, as I said, a lot of molecular assays. We did that, and it is clearly a non-apoptotic mechanisms. ZVAD has no effect on cell survival. We use number of knockout models to check that, and as non-apoptotic cell death mechanism, there's no reactive oxygen species generation. We did a bunch of toxicity studies and bunch of efficacy studies, which shows that it's very efficacious, non-toxic compound. There's some solubility issues that we are trying to tackle with our formulation team, but that is a work in progress. But what is the mechanism of this one? At least this one we have a little bit more clearer idea. It is affecting the DRP1, a mitochondria, because mitochondria was swollen. If you look in mitochondria, there is a protein called DRP1. It is dynamin-related protein 1, which do fission. So mitochondria is in constant fission fusion, right? So this is a fission protein which was being inhibited, and DRP1 has known to play a role in TNBC, but there's no such drug as potent as TPH compound that is out there. We looked into patient samples, and yes, DRP1 was super heavily upregulated, which means that DRP1 is a very important target. We started looking at its binding with DRP1. It binds very effectively. We looked at the inhibition of DRP1 with a number of molecular pathways, and we found that it is inhibiting DRP1 and its signaling pathway very effectively. We then started looking into its role in drug resistance. David and my team developed some models to look at drug resistance, and we can show that TPH class of compounds can inhibit and overcome apoptosis resistance as well as taxane resistance. And I'm happy to talk to anybody if you have interest here. The reason I'm running through this slide is just to try to show that metrophagy is another technology. This is another technology that we have developed to try to target triple-negative breast cancer. Pia Vogel is here. She works very closely in the drug resistance area, and you will hear her talk about some other drug resistance factor. We looked into other drug resistance marker, ABCG2 and ABCB1, and it is killing cancer cell through collateral sensitivity. We have done a lot of SAR studies on TPH compounds, and we are still working in this area. Okay. So it's too much. I know too many slides to cover, right? And I think I'm losing some of you in the back, so I'll stop here. So nothing is possible without the right team and right people and right guidance. So we have been working very effectively in this area, and I've developed large collaborations here. And Dr. Murthy was saying that we collaborate, and so I'm collaborating with few of the people in this room, but also outside in India. And I'm very open to collaboration. And I look for strategies to come up with newer molecules and newer ways to target cancer and drug resistance cancer, and even in other areas that we can potentially collaborate. So for example, I'm also interested in developing new tools. So we have developed some microfluidic devices. You will see some posters on that. I'm not going to talk about it. We have developed this 3D printed microscope for holographic microscopy. This is still patent pending. You can see this posters on this. We have worked on some anti-cancer drug discovery with Professor Trivedi and his team, and So lot of projects in that area that I cannot show you, but you will hear some with Dr. Karthikeyan in his talk, and then some other work. So I wanted to show you this, but I think it will take too much time. I'm very interested to see if students are interested in doing some internship in the US, and I'll be very happy to facilitate that, and we can talk about it. But I'll stop here and I'll take any questions if you have. Thank you so much.
Thank you, Professor Amit Tiwari. So it's a very good presentation. You have given one new technology of cell disintegration, that is a meiosis. I think it may be in future, it may be one of the very good druggable target. I hope so, because every day we are making new research, so and we are getting something. So definitely it will be. Yeah. So just I am from the audience. Any questions? Anybody, please?
Tick cell death leading to necrosis, and that can lead to inflammation. And in other case, it was apoptotic case. It was well-known pinocytosis, and the cells are gone. But what is the fate of the cell bodies in case of non-apoptotic cell death?
That's a very important question. I don't know if you understood the question in the back or not. So, methodology में मैंने आपको समझाया कि we got a drug, class of drug that is doing a cannibalism-like effect. Cells को मजबूर कर रहा है कि अपने आप को जाके खा लो और पी लो, right? Which is called methodology. जब cells अपने आप को खा पी रहे हैं या जो भी कर रहे हैं और उसके बाद burst हो रहे हैं, so what is happening to those debris, right? And fortunately, I put a slide on that. क्योंकि मुझे पता था कि I will get some professors like Professor Babu, which will ask me some difficult questions. And where is it? I hope I have that. Yeah. Light is on, but you can see that when the cells are dying, it's releasing some immunogenic markers. So it's an immunogenic cell death. So for example, especially with TPH compounds, which is doing necroptosis like cell death, when they're bursting and dying, they're releasing immunogenic cell death marker. And necroptosis is a well-known immunogenic cell death, right? So it is actually further killing the cancer cell death through immunogenic mechanism. And we published this finding recently. And however, we do not really know overall fate of methodophagy inducers, and we have yet to see all those effect in animal studies. I mean, cells we can show a lot of things, right? But animal studies will tell us if there are some side effects or some late developmental effects or not. So we did some early toxicity studies and subacute toxicity studies, and it's pretty safe so far, but we do not know in the long term of the effect. Thank you.
Thank you very much for answering my question. By the way, I'm not expert in this area. My question was just curiosity, what is going to happen?
Oh no, this is a very, very important question, honestly. Thank you.
So due to time constraints, last questions.
Yeah, sure.
Thank you, Dr. Tiwari. It was really nice talk. So my question is like, did you observe any any effect with the normal cells?
Yeah. So that is. Yeah, that is first thing we do, right?
After treatment with your compounds?
Yeah, sure, certainly. We do that all the time. Without that, you cannot really tell whether they are safe or not, right? So I looked into a bunch of different cells, normal cell models, and you do see methodophagy in them, but at a very high concentration. So there is a very nice concentration range why they're effective in certain specific cancer cells. They're not effective in all the cancer cells also. So we looked at many different types of cancer cells, and some they are very promising, especially triple-negative breast cancer, pancreatic cancer, colorectal cancer, brain cancer. They're very effective. Some other cancers we do not see that much, right? But as we looked at, CL1459 data that I showed you, we looked at animal studies, we did heavy rodent studies, mouse studies, and then we looked at zebrafish models. So we do a bunch of toxicity studies to really see whether they're safe, effective or not.
I mean, how this compound is?
Why is it? The question should be, why is it so effective in cancer cells versus normal cells? Right? We don't know. But our hypothesis is it's targeting some of the the basic metabolic pathways, that is where the cancer cells is differentiated with the normal cells. And these lysosome to nucleus homeostasis is very, very well developed in cancer cells compared to normal cells. There's a lot of macropinocytosis and mitosis goes in the cancer cells. Macropinocytosis goes in the cancer cells versus normal cells. So we are seeing that difference, and it is catching, same hallmark of cancer that we always talk about. What is the hallmarks of cancer? And if you can really develop drugs to target those hallmarks, I think you have to. So I think that's what it is doing, but there are some specific targets that we think is being affected, which is highly upregulated in cancer cells. For example, PIK5. PIK5 leads to, and these are actually inhibiting PIK5, and these are PIK5 inhibitors, but not very specific because they're also targeting you know that there are drugs with multi-tyrosine kinase inhibitors in the market, and they're very effective, right? So it is a multi-target inhibitor so far, but hopefully we will have more specific inhibitors which can do something very similar.
Thank you.
Thank you. Yeah, very good questions.
Yes. Sir, any metabolic study of the presented compounds? Because this compound containing the amine or CYP base, so there is some maybe chances to metabolite in acidic condition.
Yeah, we are actually doing a lot of metabolic studies of these class of compounds. Looking at different mitochondrial factors, and other factors. Metabolics. When we talk about metabolism, we're talking about a lot of different things, right? So we looked at number of amino acids, for example. If it deprivate the cells with those amino acids in presence and absence of these bad compound, what is happening? So we have some idea that it is affecting some metabolic pathways for specific amino acids, but we don't have a clear idea.
Because the compounds containing the amine portion, amine part, because it may be degraded in the acidic condition.
Yeah. There is Karthikeyan, he's an expert on that. That exact question he's an expert on. But he's sitting in the back. He will definitely answer you that question, because we discussed that question day in and day out. Right? So we sat like multiple nights just addressing some of those questions with Professor Trivedi and Karthik. Exactly the same problems, right? I mean, how to change those to affect those. But thank you so much. I appreciate your questions. Thank you so much again.
Thank you, Professor Amit Tiwari. It's a wonderful presentation, giving more information to everybody, budding researchers in this field. So more informative, and thank you.
Thank you, sir. Thank you so much for your words of wisdom. It will be a great pleasure for us to listen to you again in our future events. Now, I would request Dr. Rizwan Khan to present the memento to Dr. Murthy, the session chair, and Dr. Tiwari, the keynote speaker, as a sign of gratitude.